Hello, everybody. Welcome. This is our last video for the digestive system. I know it's been a lot. It is a large system, so hopefully you're making it through. Just as we're finishing things up, just to say, send questions, stop by a Zoom office hour. Make sure you're hitting all the anatomy, the parts and pieces and stuff from your lab manual, from your textbook. Quiz yourself, draw pictures, whatever you need to do to make sure you're comfortable with the anatomy, and then applying all the physiological concepts. All right, so this last one, they don't quite go together, but I didn't want to do two separate videos, one for each one. So we're going to do hormones first, and then we'll talk about absorption. So hormones, basically the idea of the hormonal coordination of digestion and absorption. So we talked a little bit about the neural parts with some of those reflexes. We have the gastroenteric, the enterogastric, the gastroileal. We have some of the local responses we've seen with stretch receptors and pH changes and triggering the release of enzymes and triggering the release of other like neural responses. So now we're going to focus a little bit on the hormones. So in the notes are kind of all listed, the name, when it's like what triggers its release, and you'll notice almost all of them are triggered when chyme comes into the duodenum. They almost all come from the duodenum. They have multiple targets and different effects though, so that's probably going to be the biggest part, trying to remember which does what. So there's a couple diagrams. This is from your text. I believe this is an older picture, but it's probably modified a little bit by now. I did post a PDF of scans of older pages that I'm going to show here. So if you like the table I'm going to show you or another more anatomical picture representation of all these arrows, you can find those on Blackboard. It'll just say hormones or digestive hormones or something like that. You'll see that available also. So I'm just going to go through the list that's here in the notes and kind of walk through them with this flow chart, and then we'll see the table, and then we'll wrap up with the absorption piece. All right, so the first one in the notes is secretin. So secretin comes from the duodenum. So most of these are duodenal again. So secretin, this guy right here, a lot of times they combo it with CCK because they're both released with the same trigger. Chyme, the stretch receptors, chyme coming into the duodenum triggers release of CCK and secretin, and they have similar targets, but they have different effects. So I wanted to talk about each one of those. So we do know that it's not shown in this picture, unfortunately, but we first saw CCK and secretin back in the intestinal phase of the gastric activity, in that they both inhibit the stomach's activity. So CCK and secretin inhibit gastric activity. In this diagram, they're talking primarily with the accessory structures. So secretin triggers the pancreas to release buffers. Right? CCK is going to be for enzymes. And then secretin stimulates, let me take a look, stimulates the liver for bile production. It's not really said here, so I'll say bile secretion. So that's to the liver. So that's the job of secretin. For CCK, CCK communicates to the pancreas to release enzymes. And then CCK to the gallbladder is a contraction of the gallbladder. And it also triggers the opening of the hepatopancreatic sphincter. So both secretin and CCK have an effect on the pancreas and the release of bile, kind of in their own way. So that's secretin and CCK. GIP, the gastric inhibitory peptide, we can kind of guess what it's going to do. GIP is released with chyme coming into the duodenum. It inhibits the release of gastrin from the stomach, so it inhibits gastric activity. It also targets the pancreas to release insulin. So in addition to just the blood glucose levels going up, GIP also triggers the release of insulin from the pancreas. And then we have VIP, GIP and VIP. So VIP stands for vasoinhibitory peptide. Sorry, got it mixed up. Vasoactive intestinal peptide. Vasoactive intestinal peptide again releases when chyme comes into the duodenum. And what it does is it goes and targets your intestinal capillaries, triggering them to dilate. So they open up, which makes sense because here is chyme coming into the duodenum. We know nutrients are going to be absorbed. So now we're going to dilate those capillaries, which will make it easier and more efficient for the nutrients to be absorbed. So we can see that helps to facilitate nutrient absorption. So that's VIP. And gastrin, we saw gastrin coming from both the stomach and the small intestine. So in the gastrin from the small intestine, from the stomach, we can see that it is a stimulatory hormone. It stimulates the acid production and gastric motility. It also does come from the duodenum, but in a special case. So it's not the stretch receptors, that's not just chyme coming in, but it's chyme that's coming in that is unfinished in digestion. So maybe you have large carbohydrates coming in or large protein chains coming in. And so what it will do is it will almost counter the inhibitory phase of the intestinal phase of gastric activity to try to keep some of the stomach action going. So they're kind of working almost antagonistically. Gastrin from the duodenum and GIP from the duodenum kind of are battling out. So whoever is stronger will get their signal kind of sent out a little bit more efficiently. And then there's one more that's on the list that's not on this picture. It's called enterocrinin. This goes and triggers duodenal glands to secrete their alkaline mucus when chyme comes into the duodenum. So we have this flow chart of the hormones. I have posted this table. So if you like the table, I know my head's kind of cutting off some of the effects, but you have access to this on the Blackboard. So you can see it lists all of our hormones, CCK, GIP, gastrin, secretin, vasoactive, intestinal peptide, and I had to write an enterocrinin because it wasn't in the table. And then you'll see almost all of them, arrival of chyme, 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 chyme enters the duodenum. So their stimulus is the same. Almost all of their origins come from the duodenum, with the exception of gastrin from the stomach. But here's where they differ, their targets and their effects. So probably this part of the table is what's going to be where you want to spend most of your time, because the stimulus and the origin are very similar for most of them. And then I also have this picture available on Blackboard, kind of the table and this picture go together as one PDF document. So it's just like the flow chart, but with actual organ pictures instead of just boxes. So if you like either one of those, you can use either one of those to practice and work through your hormones. All right, the last piece that we're going to cover is the big picture. We've got to our big picture. We made it. So now we've gone through all of the parts and pieces. Or you've gone through the parts and pieces. I've gone through the physiology parts with you, but now we can track those three major macromolecules that we use for nutrition, carbohydrates, lipids, and proteins. And so now we can track through all of the alimentary canal what happens to these macromolecules as they go from their large food stuff down all the way into the absorbing into your tissues, or at least into your blood and your lymph. So a lot of times you might go through an activity, or I think in a lab question might say, follow all of the nutrients in a ham sandwich. So that would be like bread would be your carbohydrates, lipids would come from the meat, and the proteins would come from the cheese or the meat or whatever. So if we're to do that, we can say, okay, so the bread is a large starch. So it's going to go in your mouth, you're going to chew, chew, chew. Salivary glands are going to release your salivary amylase. So that's the first attack on carbohydrates in the oral cavity. Swallow, it goes down into the esophagus. In the stomach, it doesn't really do much. The salivary amylase can keep working, but your mouth is a pH 7. Your stomach is a pH 2. The amylase is going to be denatured, and it denatures about pH 4 or 5. So as the stomach pH goes down, 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 the salivary amylase is going to be denatured and carbohydrate digestion stops. But you might have some disaccharides or trisaccharides by that time. Okay, so now we get into the small intestine. Buffer is released. We neutralize the chyme that's coming from the stomach. We release your pancreatic amylase right here. And now we can take our large starches, any oligosaccharides, trisaccharides into disaccharides. And so now we have our small di and trisaccharides. And then lastly, we're here at the brush border, right? So remember, the brush border was the enzymes that are found at the top of those microvilli. So here's our enzymes. Lactase breaks down lactose. Maltase breaks down maltose. Sucrase breaks down sucrose. And now we have individual monosaccharides They are brought into the cell via facilitated diffusion and co-transport. So here's our monosaccharides, and then we use facilitated diffusion to get them into the bloodstream. So there we go. We've gone from your ham sandwich piece of bread through your entire digestive tract to get to that big picture goal of absorbing monosaccharides, the glucose, into the bloodstream. All right, I'm going to skip lipids and let's do proteins because they follow a similar path, not exactly the same. So proteins, we'll do blue here. So there's no enzymatic activity in the oral cavity. Proteins just stay proteins pretty much until they get to the stomach. So now here we have pepsin. Remember, pepsin was pepsinogen that got activated by the hydrochloric acid. Large proteins get broken down into your smaller polypeptides. Now into the duodenum, neutralized by the buffer coming from the pancreas. And we had all of those, what I just called peptidases or proteases. This is just your collection of enzymes that can break those peptide bonds into small short peptides, amino acids, and dipeptides. Remember, our dipeptides were kind of like these guys. So again, the brush border enzymes are going to break that bond and allow for facilitated diffusion and co-transport of your amino acids into your bloodstream. Okay, so carbs and amino acids follow a similar absorption path. They get broken down at various places with these various enzymes, facilitated diffusion or co-transport into that cell, the simple columnar lining cell, facilitated diffusion across the basilar surface, absorbed into the capillary. So that's all going into the venous blood, heading via the hepatic portal vein, will go to the liver for processing. The sugar can be stored, the amino acids can be stored or used to make mostly plasma proteins. All right, now lastly, we have our lipids. So lipids, we have a little bit of digestion happening with our lingual lipase. So that comes from the tongue. Nothing happens in the stomach. It's going to get denatured by the low pH. So now we are in the small intestine, right? So we've made it through the stomach. Now we're in the duodenum. Here's our bile is coming from the liver. That helps to emulsify, so increasing the surface area of the fat droplets. And now we have pancreatic lipase coming in, breaking those large fats, your triglycerides, into monoglycerides and fatty acids, putting them in a micelle. So a micelle is like a ball of fat, like this, with their little fatty acid chain tails kind of sticking towards the inside, hydrophilic outside, hydrophobic inside. So those are the micelles. They are lipid soluble, so they can just do a straight diffusion. They don't need facilitated diffusion or co-transport because they're lipids. They can go through that hydrophobic phospholipid bilayer. So while it's in these cells, they kind of process them, hook them back into triglycerides, and then combine them into these large complex structures called chylomicrons. I mentioned that a little bit on one of the other videos. It's the fat triglycerides, it's some proteins, phospholipids, some cholesterol, kind of all crammed together. They're large, so they have to do exocytosis, and they get picked up in the lymph, the lacteal. So the lipids actually don't go directly into the blood, they go into the lymph first, and then from the lymph, your cisterna chyli, thoracic duct, left subclavian vein, into the general circulation, superior vena cava, heart, out to the lungs, back to the heart, then back out to the body before those lipids can be distributed out to your liver and your adipose tissue and things like that. All right, so that's our big picture takeaway for our whole system. This is the reason why we are doing the digestive system, to get monosaccharides, amino acids, and fats. My Instant Pot is ready to go. You can hear it. My rice is done. That's funny. I got some carbohydrates I'm going to consume tonight and break them down with my salivary and pancreatic amylase and absorb it into my bloodstream to go to my liver to store for later. Isn't that a good application, right? Do what you can. All right, so that's digestion and absorption of our nutrients. I want to talk one more slide about our water absorption. So we have this kind of a cool, it's pretty amazing how much water we take in and put into our tube of our digestive tract and how much we reclaim and how much is lost. It's amazing. So track my math with me here. So this is all daily values, so not just in one sitting, obviously. So in one day, we average about 2 liters that we take in. Okay, so we're drinking our water with food too, so fruits and watery foods and things like that. So we take that in. We produce about 1.5 liters of saliva every day, so that's liquid that's going into our tube. About the same amount of gastric secretions every day. Bile, about a liter, pancreatic juice, about 1 liter, and intestinal secretions, about 2 liters. So if we add all of that up, by the time we get to the intestine, we have like 9 liters of fluid that we've put into this tube. Now, clearly, we don't excrete 9 liters of fluid out of the other end, which would be insane. We'd have to constantly be drinking water. So if we take a look, we have some reabsorption taking place in the small intestine, and this is a huge amount. We talk about the large intestine reabsorbing water, and it does a really good job. But look, we go from 9 liters at the beginning of the small intestine due to all of these secretions, and we end up in the beginning of the large intestine at 1.2 liters. So that's like 7.8 liters of water that's reabsorbed along the small intestine. We dump a lot in the small intestine, but we take a lot of it back. And then from there, we gain a little bit more with all the mucus that's being secreted in the large intestine. We kind of bump up a little bit to 1,400 mils. But then we reabsorb. And by the time we poop it out, our feces contain so the 9+ liters of water that we put into the tube, our feces in a normal, regular fecal matter, not diarrhea or anything, we're losing only 150 mils of water. That's amazing. That's such a good, efficient water reclamation process we have there. So then you can also see when we're talking about diarrhea, if the peristalsis of the large intestine is too fast, or even along the entire tract is too fast, you lose a lot of water. So you can see how chronic diarrhea or severe diarrhea, even after a couple days, can be very threatening to your water balance. You can get dehydration. Cholera is an infectious disease caused by a bacteria that prevents the water being reclaimed by keeping open salt channels, and salt just dumps into your intestines. Water follows salt. Not only do you not reclaim it, you're actually losing more water out of your body, and so people can die in a couple days from dehydration because they're just pouring out all of this water through their watery feces, this watery diarrhea, which can also impact our pH, as we'll be seeing in our chapter 27, the acid-base balance. Is not only are we losing water, but remember, we have a lot of buffer in that pancreatic juice to buffer the chyme. And so if we're diarrheaing out all of our buffer, that's going to affect our blood's pH. And so this will be something else we talked about. We talked about vomiting causing metabolic alkalosis. Well, diarrhea can cause metabolic acidosis because you're losing a lot of your buffer. All right, so that's it. We made it. Whew, that is a big chapter. If we were to be in class, that would take 4 days of lecture. That's the only system I ever lecture 4 days on. It's a pretty hefty one. So hope you're doing okay. I miss you guys. We will, next, after the digestive system, we're going to get into the urinary system. So please make sure you are getting help when you need it. Send an email. My office hours are kind of lonely, so come and visit me. Just pop in, say hi, even if you don't have a question, and get some help, or just say hi. That's totally cool. All right, I will see you later. Have a good day.